So now today we are going to discuss regarding the RNA world. Okay. So coming to this RNA world, for foregoing discussion, an immediate question becomes evident. What is the first genetic material? It shall be discussed in detail in the chapter on chemical evolution. So, but briefly we shall highlight some of the facts and points. RNA was the first genetic material. There is now enough evidence to suggest this essential life processes such as metabolism. Like metabolism, we call it as defining property of living when it comes to the living world. And next we have the translation process. And we have the splicing. Metabolism, translation, splicing evolved around RNA. RNA is used to act as a genetic material as well as a catalyst. There are some important catalyst is in the sense we have ribozyme. There are some important biochemical reactions in living system that are catalyzed by RNA catalyst. So not by protein, but RNA being catalyst was reactive and hence unstable. So usually enzymes will not stay stable. It will change into a transient stage and it will come back. Therefore, DNA has evolved from RNA with chemical modification that make it more stable. So DNA being double stranded and having complementary strand further resists the change by evolving a process of repair. So in this chapter we are going to see this is double stranded. So if you see adenine is pairing with thymine, guanine is pairing with cytosine, thymine is pairing with adenine. So like this we are going to see the complementary base pairing. If one is 5 prime to 3 prime and other one is 3 prime to 5 prime. Okay. So which is anti-parallel in nature. Coming to the process of replication. In the process of replication, which is proposed while proposing the double helical structure for DNA, Watson and Crick has immediately proposed a scheme for replication of DNA. So the double helical structure of DNA is proposed by Watson and Crick from the data collected from the data of X-ray diffraction study who has done, that is Rusland, Franklin and Wilkins, and has immediately proposed a scheme for replication of DNA, means how the DNA will be replicated. So if you see here, so here you have the double stranded DNA. Initially it is unwinded. So after unwinding, what it will happen is usually the new DNA complementary base pairing will be formed to the two ends. Now this two ends consists of, it is semi-conservative method because half of the DNA is mother DNA and remaining half is newly formed. Here also half of the DNA is mother, remaining new is newly formed. In this way it is semi-conservative in nature. So Watson and Crick model for semi-conservative DNA replication. So it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for DNA genetic material. So Watson and Crick 1953. The scheme suggested that the two strands would be separate and acts as a template for the synthesis of new complementary strands. So here the two strands got separated and each one will act as a complementary for newly forming strand. So each DNA molecule, after completion of DNA molecule, each DNA molecule would have one parental and one newly synthesized one. So this scheme we call it as semi-conservative DNA replication. Experimental proof that it is semi-conservative method. It is now proven that DNA replicates semi-conservatively. So it was shown first in E. coli and subsequently in higher organisms. Remember when it comes to E. coli, the number of nitrogenous bases is 4.6 into 10 to the power of 6 base pairs and coming to the length 1.36 mm. And you have to remind the remaining one also, that is 5174. So for this bacteria, phase, then this would be, so 5386. 5386 bacteriophages. 5386 base pairs. It was first shown in E. coli and subsequently in higher organisms. So, such as, this is very important question. The experimental proof of semi-conservative method is first seen in E. coli. So, and human cell. Matthew Michelson and Franklin Stahl. Matthew Michelson and Franklin Stahl performed the following experiment in 1958. So, they grew E. coli in the medium containing N15, NH4, so N15 isotope. So, here N15 is the heavy isotope of the nitrogen and as the only nitrogen source for many generations. So, the resultant will be N15 was incorporated into newly synthesized DNA as well as the other nitrogen containing compounds. This heavy DNA could be distinguished from normal DNA by centrifuging in cesium chloride. This is also very important. And the density gradient, please note that N15 is not a radioactive isotope and it can be separated from N14 only on the basis of densities. So then they transferred the cell into the medium with normal N14 isotope and take the samples at various different time intervals as the cells are multiplied. So, and extracted the DNA that remained as double-stranded helix. The various samples are separated independently on cesium chloride gradient to measure the densities of the DNA. So, here if you see the picture, can you recall what centrifugal force is? Think why a molecule with higher mass would sediment faster. So, because its weight will be more. So, due to gravity, it will come down faster. So, here they grown this bacteria in N15. Okay. Now, they transferred this to N14. So, in the first generation, they got the hybrids. And in the next generation also, they got the pure forms of N14 and hybrids again. So, in this way, they are actually getting the maintaining of N15 for generations to generation. So, in this way, they came to know it is semi-conservative. This is after 20 minutes. After 40 minutes, this is the resultant. Here, this is the centrifugation tube. Here you have the N15. Red is denoting the heavier isotope. So, only N15 is there. Here you have the hybrid N14 and N15. Here you have the lighter isotope N14 and N14 and 15. So, this is a heavy isotope. This is hybrid and this is lighter and hybrid one. So, thus the DNA was extracted from the culture one generation after the transfer from N15 to N14 medium. That is after 20 minutes. E. coli divides in 20 minutes and has a hybrid or intermediate density. So, DNA extracted from the culture after another generation, thus after 40 minutes the second generation was composed of equal amount of hybrid DNA and a light DNA. So, if E. coli allowed to grow for 80 minutes, then what would be the proportion of light and hybrid density of DNA molecule? So, very similar experiment using radioactive thymidine to detect the distribution of newly synthesized DNA was performed in Vicia faba. So, Vicia faba, which is faba beans, by Taylor. So, and his colleagues in 1958. So, this experiment is proved that DNA in chromosomes also replicate semi-conservatively. They have taken thymine as a isotope. So, let's see the machinery here. So, coming to the machinery, in living cells such as E. coli, the process of replication requires a set of catalysts or enzymes. The main enzyme here is DNA dependent DNA polymerase, since it uses the DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes that they have to catalyze the polymerization of large number of nucleotides in very short time. So E. coli will be having 4.6 into 10 to the power of 6 base pairs. Compared to the human, it is 6.6 into 10 to the power of 9 base pairs. So the entire process of replication within 18 minutes. So the E. coli will complete the replication within 18 minutes. That means the average rate of polymerization was 2000 base pairs per second in E. coli. So for 4.6 into 10 to the power of 6, it took 18 minutes, it would be around 20,000 base pairs per second. Not only do the polymerases have to be fast, but they also have to catalyze the reaction at a high degree accuracy. Any mistake during replication would result in mutation. Furthermore, energetically replication is very expensive process. So deoxyribonucleoside triphosphates serve the dual purpose. It acts as a substrate. They provide energy for polymerization reaction. And the two terminal phosphates in the deoxynucleoside triphosphate are high energy phosphate. Same in case of ATP. Means they act as an energy supplying compound at the same time as a substrate. In addition to DNA dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated in the entire length due to very high energy requirement, the replication occurs with the small opening of the DNA helix. We call that as replication fork. The DNA dependent DNA polymerase catalyzes the polymerization only in one direction, that is only in 5' to 3' direction. This creates some additional complications at the replication fork. Consequently, on one strand, the template with polarity 3' to 5'. So the replication is continuous. While on the other strand, the template with 5' to 3' polarity, this is discontinuous. So the discontinuously synthesized fragments are later joined by the enzyme called DNA ligase. So here we got two enzymes. One is DNA dependent DNA polymerase and DNA ligase. So DNA polymerase on their own cannot initiate the process of replication, and also the replication does not initiate randomly at any place of DNA. And there is a definite region in the E. coli where the replication originates. Such region we call it as origin of replication. It is because for the requirement of origin of replication that the piece of DNA, if needed to be propagated during recombinant DNA procedures. So require a vector. The vector provides the origin of replication. So further, not every detail of replication is understood well. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division cycle should be highly coordinated. A failure in the cell division after DNA replication results in polyploidy condition. Usually it's a chromosomal anomaly. It is usually seen in plants. So you learn the detailed name of origin and process occurring in higher classes. So this is 5' to 3'. So here always this is continuous strand. This is discontinuous strand. Always the formation product will be 5' to 3', towards 5' to 3'. Template will be 3' to 5', 3' to 5'. So two enzymes we have seen. One is DNA polymerase. That too, they will start the replication at origin of replication only. And we have this for the discontinuous like Okazaki fragments, the ligase enzyme will be combining to form a continuous strand. Okay, so this is about the replication process.